Okay, so we'll start in just 1 or 2 more minutes. If anyone doesn't want to be recorded, please keep yourself without cameras, or let us know. I think we are still having some people joining in. We're at the moment with 33 participants. Okay, it's coming up, the number. Mhm. I'll give 2 more minutes before we start. Okay. Maybe I will start with the introductions and the etiquette. So first of all, thank you all for joining in for today's seminar talk. So I have the pleasure to have Professor Simon Krattinger from the King Abdullah University of Science and Technology in near Jeddah in Saudi Arabia. So Simon is an associate professor of plant science, and he's the chair of the plant science program at KAUST. Simon's research uses the latest technology advances in genomics to understand wheat evolution and biotic stress resistance. So just like you to remind, please don't interrupt the talk. You have all the time in the end for questions. Either you can put your question on the chat or you can raise your hand and I'll go through your questions to make sure that everybody has who wants to ask a question will have that opportunity. So thank you so much, Simon, for accepting the prompt invitation. It's a pleasure to have you here. And yeah, the stage is yours.
Right. Thank you very much, Sonia, for an introduction and also the invitation to give a seminar this afternoon. Oops, sorry. So I have to apologize first because my talk is not going to focus on barley. It's actually going to be on wheat. But I hope I can excite you with some of the latest research that we have done on wheat genomics, and particularly understanding the history of the bread wheat D genome. Now, of course, as you all know, cereals have been cultivated for millennia, and this cultivation of cereals is deeply rooted in human culture and human history. So we can say cereal crops have shaped human cultures, and human cultures have shaped cereal crops. And here I just want to show you two examples that sort of highlight that cultural importance that cereals had and have and still have for human culture and ancient human civilizations. So what you see here on the left is probably the oldest piece of writing that we know of today. It's ancient Sumerian. It's about 5,500 years old. And it's a clay table showing the quantities of barley rations that were distributed to workers. So up here, you can see the symbol for barley. And you may or may not know that writing was primarily invented to organize the more complex lives of ancient emerging societies. So writing was invented not to note down stories, but for accounting purposes mainly. And so here on the right, you see an image of the so-called Temple of Bacchus, which is in present-day Lebanon. And this is ancient Roman, and you see this beautiful entrance column. And you see these beautiful wheat spikes, most likely durum wheat spikes, that have been carved into this entrance column of this temple. And again, this is just highlighting again the cultural importance, the historic importance that cereals have and had for ancient civilizations, but also for modern civilizations. Furthermore, we know that cereals, or the wild ancestors of cereals, have been utilized long before domestication. So here's an example of the Natufian society or civilization, which was occupying territories in modern-day Jordan. And the Natufians are a pre-agricultural society, and they have explored and utilized and possibly even managed. Can you see the presentation?
Yes,
I can.
Okay, good. That was just something in the chat. Okay, so these Natufian societies, they were utilizing wild cereal relatives and again, possibly manage them. So here what you see is a fire pit that was excavated in Jordan. And in this fire pit, scientists found these breadcrumbs. And the thing about these breadcrumbs is they're about 15,000 years old. So they predate domestication by about 4,000 years. And these breadcrumbs are made up of wild relatives of cereal crops, mainly einkorn and wild emmer wheat. And we know today that actually the pre-agricultural, pre-domestication cultivation of wild cereals or wild relatives of cereals goes back to about 20,000 years ago. And this might be important for later on in the talk. So basically, people knew how to utilize wild cereal crops. They knew how to process them, and they also knew how to manage them long before domestication. So now I'm going to talk about bread wheat, and bread wheat is quite an interesting species because bread wheat only exists in its domesticated form. There's no wild relative, or there's no wild bread wheat. And so bread wheat emerged as a succession of stepwise hybridizations of 3 wild grasses. It combines the A, B, and the D genome, so it's a hexaploid species. So the first hybridization occurred around 500,000 years ago between Triticum uratum, which is the A genome donor, and a possibly extinct population or relative of Aegilops beltoides, which contributed the B genome. And this hybridization formed tetraploid wheat, which is wild emmer and domesticated emmer and includes the pasta wheat. And then there was a second hybridization between domesticated or a domesticated tetraploid wheat and Tau grass, Eschlop's tauschayi, which contributed the D genome. And this hybridization between tetraploid wheat and tauschayi, which formed hexaploid wheat, occurred only very recently, around 8,000 to 10,000 years ago, and most likely it occurred in an ancient farmer's field. Now, when we look at this linear representation of bread wheat evolution, which you find quite common in literature, there is one problem for me, and this is that these polyploidization events and generally domestication events, they sort of represent a strong founder effect and strong genetic bottlenecks resulting in reduced genetic diversity. Again, we assume that particularly the second hybridization between tetraploid wheat and tauschayi only occurred a few times. And so this must have been an enormous genetic bottleneck. And so the question is, how could bread wheat disperse so quickly after its formation and adapt to new climatic conditions following this major genetic bottleneck? So we know that after its emergence here in the Fertile Crescent, bread wheat very rapidly dispersed along with agricultural expansion, and it reached Northern Europe, it reached the United Kingdom just a few thousand years after its domestication and its emergence in the Fertile Crescent. And of course, a few thousand years in terms of evolutionary time frame, this is nothing. This is not enough to come up with new genetic variation that would allow adaptation to new climatic conditions. Now, I'm not an evolutionary biologist, but the one thing I learned about evolutionary biology is that sort of evolvability or adaptability coincides or correlates with the availability of genetic variation. So no genetic variation means limited adaptability to new climatic conditions. So again, the question here is, how could bread wheat so rapidly expand and adapt to new climatic conditions that none of the wild relatives would face in the Fertile Crescent? And so we took a genomic approach to investigate this question. And here I'm just showing you some time frames of technology development. So when we look at cereal genomics, particularly wheat genomics, it's of course a very much technology-driven discipline. Just going back 20 years here to the good old ABI 3730, where we sequenced a couple of megabase per day, to the latest long-read DNA sequencing technologies that really allow us nowadays to sequence a wheat genome within a few days for relatively little money. So here's just a comparison. When I did my PhD a few years ago, I spent the first 2 years of my PhD pretty much assembling a contiguous sequence of about 800 kilobases, which represents about 0.005% of a wheat genome. And so just this year, within the first 5.5 months, for a comparison, we have generated 125 million kilobases of contiguous DNA sequence of bread wheat. And so that's 150,000-fold improvement compared to what we did 20 years ago. So this is not just a small change, it's actually a pretty considerable change. And this has really transformed wheat genomics and the questions we can ask in wheat genomics. So here just some milestones in wheat genome sequencing. So as you probably know, the wheat genome is actually quite big. It's 15 gigabases, which is 5 times the size of the human genome. And so 2 milestones that I want to represent here. The first one in 2018 was the publication of the first reference grade bread wheat genome from the International Wheat Genome Sequencing Consortium. This was the Chinese Spring reference sequence. And then in 2020, there was the 10 wheat genomes project that was published, which was sort of a pan-genome, or the first pan-genome approach, where the high-quality assemblies of 10 early bread wheat cultivars were released. So back then, the major challenge really was, it was a technological challenge. How do we sequence a wheat genome? And again, this has been, to a certain degree, solved with the latest PacBio hi-fi technologies. So it's no longer a question how we sequence a genome or a wheat genome, but the question nowadays is which wheat genomes should we sequence, which of the hundreds of thousands of accessions that we can access actually in gene banks should we sequence in order to maximize biological insight. And again, In our research group, recent project, we focus on the question of genetic diversity, particularly looking at the bread wheat D genome. So what these genomic studies found actually is that introgressions played a very important role in counteracting these genetic bottlenecks that were associated with domestication and the polyploid origin of bread wheat. So what you see here on the left, these are 3 chromosomes from bread wheat, from different bread wheat cultivars from the 10 wheat genome project that I just mentioned. And here in green, yellowish, you can see bits and pieces of genomic segments that are not bread wheat. So this one here, for example, came in from Triticum timophavi. Here you see a segment that came in from tall wheatgrass, which is Tanopyrum ponticum. And so you see that these introgressions into bread wheat are actually sometimes quite large, and there are a lot of them. So it's estimated that about a third of the bread wheat genome is actually not bread wheat, or up to a third. And we saw a similar picture for einkorn wheat, which is a diploid relative of bread wheat. So when we looked at the diploid, the domesticated einkorn bread wheat gene pool, we found actually regions of increased genetic variability. So again, highlighted here as these yellow and green segments. And we found that these segments were an introgression that came in from a different wild einkorn population. So einkorn wheat, again, einkorn is a diploid relative of bread wheat. Einkorn wheat was domesticated here in southeastern Turkey from a very particular wild population called beta. And then as einkorn cultivation expanded from this region, there was an influx of genetic material from a genetically distinct wild einkorn population, so-called gamma population, into the domesticated einkorn gene pool. And this happened here in northwestern Turkey. And this introgression was most likely very important for the further expansion and adaptation of einkorn to climatic conditions that it didn't face in southeastern Turkey. So in general, these studies show that introgressions are, of course, very important for wheat evolution, for wheat adaptation to new climatic conditions. And it's a very quick way how you can increase genetic diversity. And we can imagine that many of these introgressions occurred very early during bread wheat cultivation. You can imagine that a few thousand years ago, when bread wheat emerged, wheat fields were not as homogeneous as they are today. There were no monocultures. There were different wheat species grown in the same field, most likely in the vicinity of wild relatives. And although being an inbreeding species, of course, this fostered the exchange of genetic material. And so for the rest of the talk, I'm going to now focus on the bread wheat D genome. Again, this bread wheat D genome was contributed by Tausch goldgrass, Aegilops tauschii, and tauschii actually falls into 3 lineages, which we call lineage 1, lineage 2, and lineage 3. And so here we are looking at data that were generated in the Open Wild Wheat Consortium, which is very friendly and bottom-up consortium that sequenced, wants to sequence and does sequence wild relatives of wheat. And so through this consortium, we got access to whole genome sequencing data of a large diversity panel of tauschii. And so the bread wheat D genome, if you go to literature, was contributed, or literature tells us is that it was contributed by an Aegilops tauschii falling into this lineage 2 from the southern Caspian Sea. So bread wheat emerged most likely here at the southern shores of the Caspian Sea in what is modern-day Iran, from an Aegilops tauschii population that belonged to this blue lineage 2. What's quite interesting and really important for my talk is this orange lineage here, this lineage 3, which is quite peculiar because lineage 3 is genetically distinct from the other 2 lineages, but it's geographically very restricted to modern-day Georgia. So it's only found in modern-day Georgia. And so here we further looked at the phylogeny of Aegilops tauschii. And again, we see the 3 lineages. We further split those 3 lineages into subpopulations. So for example, lineage 2 forms 3 subpopulations. And here in dark and lighter blue, these are the lineage 2E. This is the Southern Caspian lineage that contributed the breadwheat D genome according to literature. And here is again this very peculiar, genetically distinct and geographically restricted lineage 3. Now this is data from a publication from Brandy Wolf's group, the first Open Wild Wheat Consortium publication, where they actually looked at the 10 wheat genomes lines, or the lines that were sequenced in the 10 wheat genomes project, and they could confirm in their publication that actually the vast majority of the breadwheat D genome indeed originates from the Southern Caspian lineage 2, but they also found bits and pieces of L3. And again, this is interesting because, again, literature tells us that breadwheat was emerging here at the Southern Caspian shores, and the lineage 3 only exists in modern day Georgia. So these segments here in modern breadwheat, these L3 segments, suggest a second hybridization or influx of genetic material from L3 into the breadwheat gene pool. And so modern day breadwheat cultivars carry around 1% of L3 integrations. And we can probably compare this to the Neanderthal genetic footprint in the human genome, which is, as you may know, also 1 or 2% of our DNA is coming from Neanderthals. So again, L3 is quite a distinct and different subpopulation of Taoxian compared to L2. Now, if this is true, if this L3 segments in modern breadwheat, this 1% of L3 segments represent a second and independent hybridization, we argue that there should be breadwheat land races out there that have a higher content of L3, more than 1%, and possibly reflect a more ancestral state of these hybridizations. Now, in order to find them, we created what we call the missing link finder pipeline. And this is work done by a PhD student in my group, Emil, a bioinformatician. So the idea we came up with was to compare k-mer sets. So looking at small strings of nucleotide sequences. So we compared two sets of k-mers. We had, on one hand, k-mers that were specific and generated from Aegyptus Taoxian L3. And we compared these L3 specific k-mer sets to what we call the sample k-mer sets. These are k-mers generated from breadwheat accessions and breadwheat land races. And so we took a genotyping data set consisting of 80,000 genotyped accessions. This is the CIMIT collection that has been genotyped using DART-seq. So it's the advantage of this is it's a very large data set. So we could look at 80,000 wheat accessions. I was a bit skeptical when we started this because the genotyping data set here, this DART-seq or genotyping by sequencing, produces quite patchy genotyping data sets. So I wasn't really convinced that that worked. And again, the idea here is that we would compare these two k-mer data sets, and the bigger the overlap, the bigger the proportion of L3. At least that's the hypothesis. So Emil went away, and 2 weeks later, he came back with this figure here, which I have to admit, it's probably not the most beautiful figure I ever produced, but it's definitely one of the most exciting figures that I think we have produced in my career. So what you see here is each dot represents a hexaploid breadwheat land race, and colors indicate this overlap between the L3 specific k-mer set and the sample k-mer set. Blue means average proportion of L3, and the more green and yellow, that indicates higher proportions of L3. And what is beautiful about it is it really confirmed our hypothesis. So again, here is the Southern Caspian. Here is where breadwheat was originating or domesticated. And you see here that breadwheat land races collected from the Southern Caspians have a normal or a low proportion of L3. And as breadwheat expanded from this region and reached Georgia, there must have been an influx of L3 because breadwheat land races in Georgia have a very high, or some of them have a higher content of Aegyptus Taoxian L3. And so this beautifully confirms our hypothesis. And then there was a second wave of expansion out of Georgia. And as you can see, the further we move from Georgia, the lower the L3 content becomes. And this fits, of course, with the model of L3 no longer being present and recombination, again, with L2 and other bread wheat accessions. So what is beautiful about this figure, in my view, is really that this is the first time that we can probably really track the dynamics of these integrations and possibly identify land races that have a higher proportion of a certain integration. And L3 was really a model for this because, again, the L3, the Egypt's L3 population is genetically distinct and geographically restricted, so we can really look into how these segments, where they came in and how they were broken down. So we took one of these bread wheat land races from Georgia that had, according to our analysis and to our missing link finder pipeline, the highest proportion of L3, and we produced a high quality reference sequence for this land race. And we find that this land race had about 7% of L3 genome compared to the 1% found in modern elite cultivars. And most notable was a 135 megabase segment that was located on chromosome 1D. So to our knowledge today, this is the biggest L3 segment present in bread wheat. It's quite interesting. This segment contains about 500 genes and is completely absent in the modern elite cultivars. When we look at bread wheat land races, we find it occasionally. It's rare, but it's present in certain land races. And of course, this now sort of raises questions about whether this was lost due to genetic drift in modern bread wheat, or whether this was actually selection against this segment. And this is, of course, something we want to follow up in terms of, what's the meaning for adaptability and possible value for breeding. But just as a summary, I think we really managed to create a pipeline hero tool that really allows us to fish out and go and look for interesting land races that have a higher proportion of certain integration, and then trace them and look at them. And we can really confirm that there are bread wheat land races still out there that have a higher proportion of certain integrations. And so this prompted us to actually, in general, better understand the bread wheat D genome and where it came from. So what we did, we went back to our Egypt's Tauschayi phylogeny, and we took high quality bread wheat genomes, broke them down into 50 kb windows, so small 50,000 base pair windows, and then using identity by state, we assigned each bread wheat genome window to one of these Egypt's Tauschayi subpopulations. And this worked very well using identity by state because, again, we are looking at only 10,000 years of evolution between the bread wheat D genome and Tauschayi. So we found a lot of segments that actually were identical by state to one of the Egypt's Tauschayi subpopulations. And this is what we found. So again, here we are looking at the geographical map. This is the southern Caspian region. And so what you see here is the color of the circles represent the respective Tauschayi subpopulation from which the bread wheat genome, 50 kb, a given 50 kb segment originated. And the size of the circle represents the proportion of the bread wheat genome that we can explain by this given subpopulation. So no surprise in what literature tells us, the majority of the bread wheat D genome can be explained by each of Tauschayi subpopulations L2, which are the L2E, which are the southern Caspian L2s. So this is what literature would tell us, that the bread wheat D genome came in from an Egypt's Tauschayi subpopulation, which belonged to the southern Caspian L2. But the interesting thing is that these two populations only explained about 75% of the bread wheat D genome. So 25%, a quarter, could not be explained by the southern Caspian Egypt's Tauschayi. But what we found is that basically all the Egypt's Tauschayi subpopulation contributed genomic segments to the bread wheat D genome, including all four L2 subpopulation, L1 and L3. And when we look at this a bit more in detail across the bread wheat D genome, so what you see here is basically this figure represented in bred wheat chromosomes. So these here are chromosomes 1D, 6D, and 7D of Chinese spring. And Chinese spring is sort of the Columbia of wheat. It's the genome that was first sequenced. And again, what you see here is that a lot of these segments are dark blue, indicating southern Caspian origin. But then you see this patchwork-like pattern, which is really interesting. And again, indicating a lot of gene flow, a lot of hybridization from different Aegilops tauschayi populations. And again, we found that at the end, all of the Aegilops tauschayi subpopulations that we could define contributed to the bred wheat D genome, indicating a lot of gene flow between tauschayi and the bred wheat D genome. Now, on top of that, again, this is Chinese spring. On top of that, we layered information from 126 bred wheat land races for which we had whole genome sequencing data. So these are alternative haplotype blocks that are not present in Chinese spring, but found in other bred wheat land races. And again, I want to draw your attention to these orange bars again, which represents the lineage 3. Because what you can see here, if you look, and again, this is information that we generated from about 126 land races that we had sequenced. And it's quite interesting because you see, if we look across these 126 bred wheat land races, you see that there are considerable proportions of the genome that can actually be spanned with these lineage 3 haplotype blocks that are still floating around in these bred wheat land races. And so this is interesting because, again, this sort of reveals a new picture of how these introgressions or the dynamics of these introgressions, because, again, the current literature told us that, you know, on average, these L3 segments or contents in modern bred wheat were really just low, about 1%. And so through our work, we now found that, you know, the maximum that we found in a single bred wheat land race is about 7%. But when we sum this up across these 126 hexaploid wheat land races, when we look at the cumulative size of these alternative L3 haplotype blocks, we can actually span 600 megabases, which is about almost 17% of the bred wheat D genome, with these alternative L3 haplotype blocks that are still floating around in the bred wheat gene pool in the land races. And again, this is all old land races, so this predates synthetic wheat breeding. So this is not the result of artificial hybridizations. This is the result of natural gene flow from L3 into the bred wheat genome, actually. And again, this is very much similar to the Neanderthal genetic footprint, and I made this comparison at the beginning, where each human being has about 1 or 2% of Neanderthal genetic DNA. But again, if you look at across the human population, there's actually a considerable proportion of the Neanderthal genome that is still floating around in the human population. And this is very much similar here with L3. And I think, again, this raises interesting questions about the value of these alternative haplotype blocks and use for breeding and adaptability. Okay, with that, I will come to the conclusion. So first of all, our work here shows that the evolution of the bred wheat D genome was much more complex than we previously thought, or what literature would tell us. There wasn't just a single Aegilops tauschayi population that contributed to the bred wheat D genome. Instead, there was recurring hybridization and gene flow, and again, this resulted in these very beautiful patchwork-like haplotype patterns that we find in the bred wheat gene pool. And this is quite interesting because the tauschayi population, actually, if you go and look at tauschayi, the tauschayi population is very well structured, and there's very little evidence for gene flow between different Aegilops tauschayi subpopulations. And so one hypothesis we have is, one possible explanation that we have is that actually the original tauschayi accession that gave rise to the bred wheat D genome might have already been admixed, and it might have already carried genomic segments from different subpopulations, and possibly this was the result of pre-domestication management of tauschayi, for which there is actually some evidence. And again, there's some interesting questions and follow-up questions regarding the cumulative length of these alternative haplotype blocks, which I think is quite interesting. And again, in the next step, we really want to dive into this and ask questions about adaptive potential and, again, benefits wheat breeding. All right, with that, I want to conclude, and I want to thank a few people. Particularly, I want to point out Emil here, who, again, a very talented PhD student in my group, who actually led this work. I want to acknowledge Andrea, Kat, and Brande for leading the Open Wild Wheat Consortium. I unfortunately cannot mention all of the members of the Open Wild Wheat Consortium. And then Jesus and Cristobal from Giannina's Center for helping us to implement the IBH pipeline, which was really instrumental here to do this haplotype analysis. It's a very beautiful pipeline that has been developed in Cristobal's lab. Also, to advertise, if you're interested in KAUST and possibly coming to KAUST, we have an open assistant professor position in plant biology. So scan the QR code and apply, please. And with this, I want to thank you for your attention and hopefully having a good discussion. So thank you very much.
Thank you so much, Simon. It was an impressive talk. Very, very interesting. I open the floor to questions. Otherwise, I will flood you with questions, and I don't want to do that. Okay, I have here the first question from Robbie. Robbie, do you want to ask instead of typing?
No, I typed because I could remember it when I was typing. Well, of course I'll ask the question. I was interested in the comments about the L3 integration that you observed in hexaploid wheat, and you mentioned this large 150 megabase integration that contained something like 2,000 genes. Now, maybe I misunderstood, but I think, well, as I recall, what you said was these 2,000 genes were absent rather in the original D genome in hexaploid wheat. Is that correct?
No. It's about 500 genes, I think. No, they're 500. Not absent, but it's
just different.
No, of course, it's just a very different haplotype block. Again, L3 is quite distinct from L2. So essentially, these 500 genes, instead of being from L2, are now from L3. We looked at presence absence, but I can't remember. I thought that something like 20% of the genes show presence absence variation in that haplotype segment, or in that particular segment between L2 and L3.
20% is high.
I think that's what I remember, yes. But it would be like 100 genes.
Obviously, I just misunderstood what you said.
But of course, then we find a lot of SNPs between the genes from the L2 segment and the L3 segments. But it's not a presence absence. It's not a presence absence variation. In modern bread wheat, you would have the synthetic region, but just from this other subpopulation of Tauschian.
Can I ask another question? I don't see any more in the text, so I'm going to ask another one.
Question.
I think right at the very beginning of the presentation, you said that 10,000 years wasn't enough time to evolve, if you like, to become adapted to different environments. Did I misunderstand that as well?
No, I said, no, I didn't say it wasn't enough time. I just said it wasn't enough time to generate new mutations, or enough new mutations to generate genetic diversity. Of course, you can look at the few mutations that it took to domesticate wheat. That was, of course, we know that these happened, but just from a general point of view of having increasing genetic diversity, I would argue that 10,000 years is not enough to counteract this enormous genetic bottleneck that was associated with the emergence of hexaploid wheat. So something like introgressions, of course, bringing a lot more genetic material and new genetic innovation in that sense than de novo mutations.
Is that unique for wheat? Because I would think that it was the opposite case in barley. Maybe other people online would like to comment.
It would be interesting to have a discussion on this, yes.
If you look at the barley genome, for example, then one of the main sources of variation is to have things like copy number variants, which are the result of directed evolution towards probably some kind of adaptation. You have selection for various things for different uses, for example. And of course, as you know, barley's adapted probably to a much wider geographic range than wheat.
Yes, sure. Yes.
It's also diploid.
So, again, I think in the case of wheat, again, it's just amazing how frequent these integrations are, and probably this is a consequence of the polyploidization. Yeah. Maybe bread wheat just tolerates a lot more integrations than barley. We haven't looked into barley. It would be interesting to do this. And it's just amazing. Again, the estimates are that about a third of the bread wheat D genome. No, of the bread wheat genome is not bread wheat.
I have another question here from Gary. Gary, do you want to ask?
Yeah, hi Simone, great talk. Really, really nice work. So I'm wondering about these L3 integressions, and you make the case that there's likely adaptive alleles housed within these particular, land races that contain various L3 integressions. Has anyone actually demonstrated that, functionally shown that there are adaptive alleles in any of those integress regions?
Maybe I was wrong. I don't know if there are adaptive alleles. I think there might be, but I don't know. Maybe this was, maybe there's a reason why there's only 1% left in the modern day elite cultivars. Maybe these were actually having a negative consequence, and that's why they have been removed or purged. I don't know. This is something we really want to look into. We haven't done this. So we don't know. We just don't know if these are, I mean, again, it brings in new genetic variation. Whether this was beneficial or detrimental or neutral, we don't know yet.
Thanks.
Okay, so we have a couple more questions. Now people lost their shyness. So is high prevalence of L3 integressions in landraces close to Georgia area related to initial hybridization event of bread wheat with L3, or does it show that integressions are more adaptive to this particular environment?
No, it must have just been I think again, so it's very important. I mean, there were integrations from other populations as well. As I showed, I think I showed that all the tau-share subpopulations contributed to the bread wheat D genome. I just focused on the L3 because it's such an easy case to study, because L3 is genetically distinct and geographically restricted. So it was very beautiful to localize it and localize the events and what happened afterwards, which is of course much more tricky if you look at something like L2, which is growing over or occurring over a wide range. So so that's why we focused on L3, just because it was the lowest hanging fruit. But we can do similar things for other integrations and go and use missing link finder to pull out land races that have higher integrations of something else if we want.
Good, thank you. Then another question. Excellent talk, Simon, from Young B. Any aging information on 3D lineage?
In terms of when it came in?
I would say that's what I would understand, yeah.
Again, the problem is we're looking at only 10,000 years of bread wheat history, and we have wrapped our heads around how we could date that integration. If anyone has an idea, please let me know, and I'll give you all the data, and you can do it if you want and publish it, because we have thought about this a lot, and we haven't come up with a clever way of how we could date when these segments came in, and it would be very interesting to do, because we don't know if this is something that happened right after, like 9,500 years ago or 2,000 years ago. It would be very interesting to figure that out.
And another question from Catherine. Are there any phenotypic differences associated with the different introgression lines which might indicate adaptation?
Excellent question. Again, this is where we need to start. This is just, by the way, very recent data that we got. So it was descriptive so far, but now we really want to look into. The problem is, of course, we're talking about bread wheat land races, and there's a lot of phenotypic variation so far, but the tricky thing is to really make sure to study if this is associated with the integration segments or not. So, and I think to do this, we will probably have to cross these segments into a common background and then really start to do some genetics on this, which we haven't done.
Another question from Robbie. Why would genetic material that comes from genetics adapted to a very specific eco-geographical region, which is Georgia, spread widely into different ecological niches?
Excellent question. I'm sure Robbie knows the answer.
If only.
Again, look, I'm looking at this as really as something, but there was a genetic bottleneck and there was a need to bring in genetic variation or create genetic variation. And again, in barley, this might have been copy number variation. In bread wheat, this might have been introgressions primarily, right? And so the question is, how these combinations, again, you bring with this 135 megabase block, that resulted in 500 genes that are quite different sequence compared to the L2. And of course, this results in all different new combinations of genetic material, and what that does, I don't know, but it's just, what we observe. So I don't know if that helps, Robbie.
I don't know the answer. I just think it's quite interesting.
I think you have to ask me in 10 or 20 years from now.
I hope soon. Can I ask you something? I was thinking about, because you started your talk with archaeological sites and all of that. So I was wondering, have you ever looked at ancient DNA from those regions and try to see if you have same signatures of the lineages on those and that ancient material?
You sort of guessed what I would like to do next.
Yeah.
We have collaborated with people from Jordan on getting ancient seeds. Unfortunately, all of what we have obtained so far was charred, and from charred grains you cannot extract DNA.
Yeah. You probably don't have waterlogged remains there, because sometimes they have
not in these waterlogged remains in these
not
regions.
in those regions. Yeah.
Of course, one thought would be environmental DNA, but again, there's challenges associated with that. So it would be very interesting, right, to go into Jordan or Georgia, for example, look into ancient grains and see, yes, because again, the maximum we find now in across these 80,000 contemporary land races is 7% of L3. I'm sure there must have been something there that had a lot more in ancient times, and it would be interesting to find that. But so far, we really have the bottleneck is unfortunately the quality of ancient seeds.
Yeah, of course. Yeah, it's a problem across the board for everyone who wants to look at more old data. Yeah. Any other questions? No. I might just wrap up with one question. So, in those regions of L3, and you said that there's different genes. If you look at the genes, and are they related with selective sweeps areas or something like that?
There's one very interesting region. If you look at that 1% that is left in the elite cultivars, there is one segment that contains a gene responsible for bread wheat quality. The publication from Jesse Poland's group a couple of years ago, where they could show that one of the superior bread wheat dough quality genes actually sits or came in from L3. So this is a very concrete example where this integration had a phenotypic consequence. And again, for the rest, we just don't know yet.
Okay, good. Thank you. I think if there are no further questions, I would probably just give a big hand of applause to Simon. Thank you so much for the lovely talk.
Thank you very much.
And yeah, thank you so much. And I hope to see you soon in the future for the International Barley Hub seminars. We'll continue with very exciting topics. Thank you.
Thanks, Simon.
Cheers. Bye.
Thanks.